The overall goal of this procedure is to label olfactory sensory neurons in vivo to visualize axonal morphology and development. This is accomplished by first electroporating fluorophore-coupled dextrans into single olfactory sensory neurons. The second step is to wait 24 hours to allow the dye to spread into the axonal processes of the sensory neurons. Next, a tadpole is anesthetized and a three-dimensional image stack of the olfactory bulb is acquired using multiphoton microscopy. The final step is to repeatedly examine the labeled neuron in the same animal after specified time intervals. Ultimately, reconstruction of cellular morphology allows the visualization of changes in axonal morphology.
This method can help answer key questions in the field of neuroscience, such as how axons grow towards their target regions in the forebrain and how synapses are formed.
Before the experiment, ensure that the electroporation setup consists of a stereo microscope with a large working distance and is equipped with fluorescent illumination and filter sets for the dye used. For electroporation, use either a dedicated single-cell electroporator or a generic square pulse generator attached to an oscilloscope. Connect the electroporator outputs to a pipette holder and a bath electrode. Ensure both the pipette holder and the bath electrode contain silver wire.